Good morning, everyone. We are from Group 2. Today we will be discussing about Shigella, a gram-negative bacterial pathogen known for causing shigellosis, a severe diarrheal disease. So this presentation will be started by myself, Erina, followed by Anmol, Mariam, and Suhair. Shigella is a gram-negative bacterium. It is non-motile, non-spore-forming, and non-capsulated. Morphologically, it appears as short, rod-shaped cell ranging from 1 to 3 micrometer in length. Biochemically, it is facultatively anaerobic, lactose-negative, and does not produce glucose. These characteristics are crucial for its identification in microbiological studies. So Shigella has four primary serogroups. So the first one is S. dysenteria. It's the most virulent, causing bloody diarrhea with fever and dehydration. We also have S. flexneri. It's the most common in developing countries, leading to moderate to severe case of shigellosis. We also have S. boydii, which is the least common, is typically isolated in India's subcontinent. And we also have S. soni, is the most prevalent in developed countries and usually is a mild and self-limiting diarrhea. So Shigella show environmental adaptability. At body temperature 37 degrees Celsius, it becomes invasive, expressing virulence plasmid like IPA-A, IPA-B, IPA-C, and IPA-D. These plasmids are essential for infection. Interestingly, at ambient temperature like 30 degrees Celsius, these plasmids are not expressed and the bacteria remains non-invasive. It is also highly acid resistant, surviving through the digestive tract at a pH as low as 4.5, and it also tolerates salts and organic acids found in fruits, vegetables, and fermented food. While freezing and refrigeration prevent growth, Shigella can still survive under this condition. So the survival mechanism of Shigella makes it a formidable pathogen. Its infection dose is alarmingly low, just 10 cells for S. dysenteria or 500 cells for S. soni can cause illness. It survives on an inanimate objects or vomit, making surface potential transmission vector. In water, it persists with minimal population decline, which explains its historical association with waterborne outbreak. Shigella can also survive in package under vacuum or modified atmosphere. Now let's assess the risk of Shigella contamination in specific food items. So for satay, it's a low risk as the high cooking temperature typically eliminates Shigella, but the contamination may occur through raw ingredients like peanut sauce. Nasi lemak is a moderate risk. Raw cucumber or improperly handled sambal could harbor Shigella. And ayam goreng beropak is a low risk due to the high temperature cooking. For kerang rebus, it's a high risk because the shellfish may harbor Shigella if harvested from contaminated water and is subsequently boiled. For chendol, it's a moderate risk because ingredients like shaved ice and coconut milk could be contaminated if prepared with unclean water. For fresh cut fruits, it's a high risk. This is because the contamination occurs during the handling of the fruits.
Good evening. My name is Anmol, and I'll continue the presentation by explaining the possible sources of contamination by Shigella species. Shigella species is a highly infectious pathogen transmitted via the fecal-oral route, causing foodborne illnesses. Contamination often occurs due to improper food handling, cross-contamination, and also environmental factors. Firstly, improper food handling includes inadequate hand hygiene, where food handlers who do not wash their hands thoroughly after using the restroom or coming into contact with contaminated materials can transfer bacteria to food, or usage of untreated water for washing produce and mishandling of raw foods. Secondly, cross-contamination arises when food handlers, using the same gloves or cleaning towels, cutting boards, or serving trays for raw and ready-to-eat foods without proper cleaning across multiple food items without sanitation can also spread Shigella. And storing raw foods near cooked or ready-to-eat items can result in bacterial transfer through contact or dripping liquids. Lastly, environmental factors include unsanitized surfaces, contaminated water sources, and factors such as flies and other insects. Improper disposal of waste and wastewater near food preparation areas also increases the risk of contamination. Therefore, effective hygiene, proper storage, and clean preparation areas are critical in minimizing this risk. Here are the common sources or reservoirs of Shigella species. Raw foods like fresh produce and raw meat are major contributors. Fresh produce such as cucumbers and fruits can become contaminated during harvesting, transportation, or washing if polluted water is used. Similarly, raw meats, particularly poultry, are susceptible to contamination from handlers or contaminated surfaces during processing and preparation. For outdoor food preparation settings, this is often seen during festivals or outside stalls which lack proper sanitation control. These environments can expose food to airborne contaminants, pests such as flies and dirty utensils, increasing the risk of bacterial transmission. Next, poor hygiene practices by food handlers or even asymptomatic carriers further contribute to contamination. Individuals with active infections or inadequate hand washing habits can easily transfer Shigella species to food, utensils, or preparation surfaces. Lastly, contaminated water is another significant reservoir. Polluted water, often used for washing produce, cooking, or as a direct ingredient, such as in making ice for desserts like chendol, can introduce Shigella species into the food chain. This is particularly concerning in areas where water treatment facilities are inadequate.
Therefore, the food items that we think is the most likely to be causing food poisoning at a festival is kerang rebus. This can be due to several reasons. First of all, cockles are often harvested from coastal water, meaning that it can be contaminated with human fecal matter carrying Shigella species. Then, cockles might not be cooked thoroughly and Shigella could survive. And even if the cockles were thoroughly cooked or boiled, improper handling after cooking could also reintroduce Shigella species. Then cockles are usually eaten in larger quantities, meaning that any contamination could expose many individuals or crowds to Shigella. And based on previous outbreaks, uncooked food which may have received extensive handling, such as prawn cocktail or tuna salad, have been implicated in a number of Shigella outbreaks.
Hello everyone, my name is Suhairmai and today I will be presenting the steps in our testing protocol. This figure represents the testing protocol for detecting Shigella that will be further explained in the next slides. First, we will begin by collecting 25 grams of each food item septically and then store the samples at 2 to 8 degrees Celsius to prevent contamination. Samples must be processed within 24 hours to ensure their viability. Next, we will enrich the samples using gram-negative or tryptic soy broth to promote the growth of Shigella while suppressing the other bacteria. Incubation is done at 37 degrees Celsius for 18 to 24 hours. This is a vital step to detect the low-level contamination. We will then isolate presumptive Shigella colonies using XLD agar, where they appear as red colonies without a black centers, and McConkey agar, showing pale, non-lactose fermenting colonies, and the incubation is 18 to 24 hours at 37 degrees Celsius. Biochemical tests will confirm the identity of Shigella. For example, the triple sugar iron test will produce a specific pattern of results, and additional tests like indole and citrate utilization will further validate the findings. Next, we will be using the molecular methods, such as PCR, to target a specific gene, which is IPA8 gene. This will provide a definitive confirmation of Shigella. We will amplify DNA using specific primers and analyze the results with gel electrophoresis. If the IPA8 gene is present, we will see a DNA band corresponding 620 base pair. This will confirm that the pathogen is present. Later on, for rapid detection, we can use ELISA. This method will quickly and sensitively identify Shigella by specific antigen. It will make it useful for outbreak situations. Finally, we assess the antimicrobial resistance using the creepy power disk diffusion method. After incubating the samples, we will measure the inhibition zones to classify the pathogen and guide the treatment option.
Hello everyone, my name is SIR2031. This table provides a comparative overview of the ISO and BAM methods for detecting pathogens in various samples, particularly in food and related industries. Let's go through the key aspects step by step. Firstly is sample types. Both methods are designed in different applications. ISO focuses on food and animal feeding samples, while BAM is more commonly applied to food and cosmetic samples. Second is pre-enrichment media. For pre-enrichment, ISO uses buffered peptone water, which helps revive the injured bacteria, whereas BAM employs lactose broth, which can also serve as an indicator media. Third is selective enrichment media. In the selective enrichment step, ISO uses gram-negative broth, while BAM uses selenide cysteine broth. Both media are designed to suppress non-target bacteria while promoting the growth of both Shigella and Salmonella. Isolation media. The isolation media varies between two methods. ISO emphasizes hectoin enteric agar and xylose lysine deoxycholate agars, while BAM includes macogni agar and XLD agars. Biochemical tests. Both methods rely on biochemical tests for confirmation, such as lysine decarboxylation urea, hydrolysis, and TSI agar. BAM additionally includes LIA agar for lysine decarboxylation testing. Serological tests. Both protocols require the use of specific slides for the agglutination to confirm the presence of target organism. Seventh is the approximate time for results. The time frame for results is similar in both methods, taking approximately 4 to 5 days. Sensitivity and specificity depend largely on the sample type and the contamination level in both methods, which means accurate results depend on careful handling and preparation samples. So this section focuses on the limitation of traditional culture methods and the potential of molecular techniques to enhance the detection of pathogens, particularly Shigella. So let's take a look. A review by NEAS in 2024 highlights the traditional culture methods, such as those specified by ISO standards, are not always efficient. They are time-consuming and may fail to detect viable but not culturable cells, which can lead to false negatives. The authors suggest that the incorporating molecular techniques such as PCR can significantly enhance the efficiency and accuracy of pathogen detection. So to sum up, PCR can overcome the limitations of culture-based methods, especially in detecting pathogens that might otherwise remain undetected. Secondly is another study conducted by Warren in 2005 provides a more direct comparison. They evaluate traditional culture methods alongside FTA filtration nested PCR for detecting Shigella. The results showed that molecular methods not only provide increased sensitivity but also reduce the time required to get the results. This suggests that combining molecular techniques with established protocols like BAM can improve detection rates and might make the entire process more reliable and faster. These findings underline the importance of evolving our detection techniques to address the growing complexity of foodborne pathogens.